Welcome to this lecture on gene therapy, a fascinating topic in animal biotechnology. Today, we're going to explore how modern science is shifting from merely treating the symptoms of a disease to actually curing it at its very root. Imagine being able to fix the fundamental genetic code of a living organism. This is not science fiction. It is happening right now in clinics around the world. Let us dive in and understand how this revolutionary medical technique works. To start, let us think about how genetic diseases actually work. Imagine you have a beautifully written recipe book, but one crucial recipe has a typo that ruins the cake every single time you try to bake it. Instead of trying to fix the bad cake with extra frosting after it is already baked, wouldn't it be much better to just erase the typo and write the correct ingredient directly into the book? This is exactly how gene therapy works in the human body. When a person has a genetic disease, their DNA has a mutation. Gene therapy acts as a molecular whiteout, delivering a correct, healthy copy of the gene directly into the patient's cells. Now that we understand the basic analogy, let us look at the formal scientific definition. In scientific terms, gene therapy is a highly advanced medical technique that treats or prevents disease by altering the genetic instructions within an individual's cells. Instead of using traditional drugs or surgery, doctors introduce a healthy, functional copy of a gene into the patient's cells. This healthy gene compensates for a mutated or missing gene. The ultimate goal of this entire process is to restore normal cellular function. By providing the correct genetic code, we ensure that the correct, life-sustaining proteins are synthesized by the body, effectively curing the disease at its source. This brings us to an important question. How do we actually get this healthy gene inside the patient's cells? We cannot just inject raw DNA and hope for the best. The delivery is usually achieved using a specialized vehicle called a vector. Most often, this vector is a modified virus. Now, hearing the word virus might sound scary, but scientists completely strip these viruses of their disease-causing abilities. What remains is the virus's natural, highly efficient ability to enter human cells and insert genetic material. So we hijack the virus's delivery mechanism to safely transport our therapeutic gene into the target cells. Now that we have our delivery vector, how is the actual procedure performed? There are two primary methods used in clinics today. The first is called in vivo gene therapy. In this approach, the viral vector carrying the healthy gene is injected directly into the patient's body, targeting the specific organs or tissues that need repair. The second method is called ex vivo gene therapy. Here, we take a more controlled approach. Doctors extract cells from the patient, genetically modify them outside the body in a laboratory setting, and then safely return these newly corrected cells back into the patient. Let us visualize these two methods clearly with this diagram. On the left side, you see the in vivo approach. Notice how the syringe containing the viral vector is directly injecting the therapeutic genes into the patient's body, where it travels to the target cells. Now, look at the right side, showing the ex vivo approach. First, cells are extracted from the patient and placed into a petri dish. The viral vector is then added to this dish, modifying the cells in a highly controlled laboratory environment. Once the cells successfully take up the new gene and multiply, they are injected back into the patient. Having seen how it works, we must understand why gene therapy matters so much in the real world. The importance of this technology cannot be overstated. For decades, patients with severe genetic disorders like cystic fibrosis or sickle cell anemia had to rely on lifelong, painful treatments that only managed their symptoms. Gene therapy offers a permanent cure. Today, it is being used to restore vision in patients with inherited blindness and to give children with severe combined immunodeficiency, often called bubble boy disease, a fully functioning immune system. It is even paving the way for revolutionary cancer treatments, like CAR T-cell therapy, which reprograms a patient's own immune cells to hunt down cancer. With such powerful treatments, precision is absolutely critical. To make gene therapy work safely and effectively, scientists cannot just guess how much of the therapeutic vector to give a patient. We must ensure enough cells receive the new gene without overwhelming and killing them with too many delivery vehicles. If we use too few vectors, the therapy will fail. If we use too many, it becomes toxic. This delicate balance requires precise mathematical calculations to determine the exact dosage of the viral vector before any treatment begins. This brings us to our first critical calculation, the multiplicity of infection, or MOI. Conceptually, this formula tells us exactly how many delivery vehicles we are assigning to each cell on average. The formula is M equals V divided by C. Here, M is the multiplicity of infection. V stands for the total number of infectious viral particles we are adding. C represents the total number of target cells we are trying to treat. If the value of V increases while C stays the same, the MOI increases, meaning each cell gets bombarded by more viruses, risking toxicity. If C increases without increasing V, the MOI drops, meaning many cells might miss out on the therapeutic gene. Let us apply this formula to a real clinical scenario. A doctor is performing ex vivo gene therapy on a young patient with severe combined immunodeficiency. The doctor has extracted 5 million bone marrow stem cells. To avoid toxicity, the medical protocol requires an MOI of exactly 3. We need to find V, the total number of viral particles required. We start by rearranging our formula using basic algebra, multiplying both sides by C to get V equals M times C. Substituting our values, we multiply 3 by 5 million. The final answer is 15 million viral particles. Physically, this means the doctor must add exactly 15 million viruses to the petri dish to ensure an average of 3 vectors interact with each stem cell. After delivering the genes, we must evaluate how well the procedure worked. This is done using the transfection efficiency formula. Conceptually, this calculates our success rate. The formula is E equals N sub T divided by N sub total, all multiplied by 100. E represents the transfection efficiency as a percentage. N sub T is the number of successfully transfected cells, meaning the cells that actually took up and are expressing the new gene. N sub total is the total number of cells we attempted to treat. A higher value for N sub T drives the efficiency closer to a perfect 100%, meaning our vector was highly effective at delivering its payload. Let us see how this works in practice by continuing our previous example. After the procedure, the doctor tests the 5 million treated cells. Laboratory results show that 4.2 million cells are now producing the correct, healthy immune system protein. We want to find the transfection efficiency. We plug our values into the formula, placing 4.2 million in the numerator and 5 million in the denominator. Because both numbers contain a factor of 10 to the power of 6, these terms neatly cancel each other out. Dividing 4.2 by 5 gives us 0.84. Multiplying by 100 gives a final answer of 84%. This high efficiency means the cells can now be safely transfused back into the patient. We have covered a lot of ground today, so let us summarize the most important ideas. First, gene therapy represents a paradigm shift in medicine, allowing us to cure genetic diseases at their root by replacing faulty DNA instructions with healthy ones. Second, we learned that modified viral vectors are essential delivery vehicles, whether the therapy is performed inside the body or in a lab. Finally, we saw that this biological process relies heavily on precise mathematics. Calculating the multiplicity of infection ensures patient safety, while measuring transfection efficiency confirms our success. You now have a solid understanding of both the biology and the mathematics behind gene therapy.